right. I'm Dr. Ben Newman. I'm a coronavirologist with lots and lots of years experience growing these dumb viruses. And I'm going to try to answer your questions. So this question is from Barb. And how are you doing, Barb? Yeah, it's been a while, but always good to hear from you. Okay, so I know you've addressed this before. Oh, I have to death. Yeah. But the theory of the SARS coronavirus 2 being lab created is once again in the news. Dumb the first time, dumb this time, dumb every time in between. Yeah. Um, can you elaborate on whether this could be true and what the genome is versus a bat virus? Yeah, sure. Oh my goodness. Yeah. So we've got pretty good reason to believe that this is a bat virus, bat origin, and I feel like even the tinfoil hat people are generally on board with that. Um, the That comes from the fact that people have found a lot of viruses that are fairly similar but not quite identical, and they're in bats for the most part. A couple in a pangolin, but it looks like the pangolins may have got it from a bat. So yeah, bats all the way down from there. Right. So the idea is that it was like engineered, which is dumb, or that it was grown up and then somebody oopsied and just sprayed it everywhere and then said, "Oh no, yeah, we can't tell anybody." Which, yeah, this is dumb. Um, yeah, we don't even need to go into all the reasons why that's dumb, but you can actually tell from the genome that this thing does not belong in humans. So the idea is that this perfect strain was either picked out or brewed up in an evil science laboratory. You got to picture that Jacob's ladder thing, with a two and the little electrical jot that goes zzz, up right in the background. So imagine that going back there, and some boiling round bottom flasks on a stand because people love those. Yeah, with some green stuff in them. Why not? Um, the virus has been changing in particular ways that tell you that it is not very, or was not very well adapted to humans, and it is now adapting well to humans. And the fact that it wasn't adapted very well to humans, I think, is a pretty good indication that, yeah, this is not a human virus, and this was not, whatever weird thing you want to imagine. You can't even track them all down. So, what are the things we've seen? We've seen big changes in that in almost every strain, a thing called open reading frame 8 gets deleted, or it loses a big chunk, or it gets a mutation in it that just stops it and doesn't get made. We know from other work on the original SARS coronavirus that open reading frame 8 is there, and it is something that helps this virus to grow when it's in bats. It is it's just a little factor that helps the virus out and hurts the bat a little bit in some way, and that's fine. It either has no function in people, or the function is not associated with virus survival, because you only spread the virus that actually gets into a cell and then gets back out again on the other side. And so that has been wrecked from very nearly the beginning. And we saw exactly the same thing with the original SARS coronavirus, which was also a bat virus. Yeah. All right. The next point is that the spike has been changing. People were saying, oh, it's a perfectly adapted spike. It turns out a lot of animals have similar ACE2 proteins, and the way these things bind, it's not exact. It's basically based on shape. So if you've got a banana-shaped thing, yeah, which actually the human receptor is banana-shaped. So here, we'll imagine that. And you got kind of a cup-shaped thing, which is what shape the spike is. Those two will sit on each other as long as the charges don't repel each other. So if there are bits that want to stick together in here, so like a generally hydrophobic amino acid next to a hydrophobic one, boom, those are going to stick. If you got a positive on this side and a negative on that side, boom, those are going to stick. And you just have to have enough of these interactions, and then, yeah, those two shapes will stick together. All the spikes have this shape. All the receptors have this shape. Most of the receptors have the same, roughly the same distribution of amino acids on that little banana-like face. And so, yeah, it's really not that, earth-shattering that you could have a virus that's able to jump from one species to another. There was just a paper that showed that white-tailed deer, yeah, can get SARS-CoV-2 and can pass it among each other. I don't think that's going to be a problem, except maybe at their deer farms, things like that, where they raise them for different reasons. And so you might see something there, like between the workers and the animals, like we saw at mink farms in various countries, because mink can get it. Yeah, cats and dogs can get it at least a little bit. A lot of animals can get it. So there we go. Yeah. Another way that we know that this is not lab created, which is goofy. So one of the original ideas was that it's the Furin cleavage site, which is about halfway down the spike. There's a little thing that has to get cut by a particular protein. And there are actually several different proteins that can cut this spot. That cutting activates the spike. So the spike doesn't work as well unless you cut that particular spot. And then once you've cut that, then another protein comes in and cuts it at a second spot. And so that's fine. Yeah. So we've seen recently that the virus has been mutating. And so something like the India variant or something like the UK variant, both of these things have expanded their furin-like cleavage site, meaning they've put in more amino acids to make it a bigger, more cleavable site. And people have done some experiments, and sure enough, yeah, it is definitely easier to activate these viruses. We're not sure yet if that makes any real difference in how well the virus grows, but you could sure see a way that it could do that, right? And we know from avian flu, from bird flu, that if you have a very small furin cleavage site, those are generally not dangerous. But as the furin site gets bigger, that's when you start to get the high pathogenicity version of the avian flu, the ones that just tear through chicken farms and kill a lot of birds and also cause terrible things to happen in people. So yeah, it looks like that. But anyway, the idea that the virus is basically building itself a better furin cleavage site, even though just out there in the world by random change, kind of tells you that this is something that can be done. And I think a lot of people think that the furin cleavage site is a really complex thing. It is, in the original version of the virus, 4 amino acids, only 2 of which are probably super important. No, it's 3. Actually, it's 3 amino acids, yeah, out of 4 are important. And we're changing that to 4 or 5 of the correct amino acid there, so for greater cleavage. Yeah, neat, huh? So that's what's going on. And once again, that shows that this was definitely not a perfectly created, ideal human bioweapon, because if you're trying to make a bioweapon, you wouldn't put in 3 out of 4. You'd put in 10 of that amino acid right in a row, yeah, to make sure it, went really well and really fast and had the greatest chance of activation. And that may not be right. That may end up being something that just causes the virus to fall apart or, be destroyed. But yeah, if you know people, generally, if you give them a gas pedal, they're going to push it as hard as they can. So yeah. So all this is, just made up nonsense. What it is a Russell's teapot argument. And so the idea there is that I think it's Bertrand Russell, philosopher, said, well, what if I were to say to you that there's teapots orbiting somewhere between the Earth and Mars in that great big space. It's too small to be seen by a telescope or any other instruments, but it's totally there. The idea is that you can't prove it's not there. The perfect little China teapot, floating way out there in space. But you can be pretty sure that it's not there because how would it get there? How would it form? Yeah, in a kiln with clay that's from Earth? And then, I don't know, yeah. And so even without being able to prove something, you can show that it's vanishingly unlikely. And therefore, you can say, you know what, this is a thing that we can't worry about, or something that we will never be able to prove, but it's also kind of irrelevant and stupid and, yeah, not really something where a lot of productive thought can be done. It's a thing you're never going to get an answer to, and I think the key is to just get over needing that answer that you're never going to get. But whatever, live your life. I'll live my life. Everybody else can live their lives how they want. Yeah, and that's fine. So yeah, for the teapot obsessed, yeah, keep hunting that origin story. Yeah, somewhere out there. I actually don't think there's any evidence in favor of it, and there's so much evidence against it that it just seems crazy to me that anybody would consider it seriously. But the idea that somebody you don't know might not be trustworthy is, I think, what's driving this. And that's pretty much baked into the way human brains work. You don't know somebody, like, I don't know about you. You may be bad news. That's a very easy thing, I think, to think. And I don't know why it's in there. I'm not going to get evolutionary psychology-ish on you, because I think that's mostly bunk too. But yeah. I could see a way. So I hope that, I don't know, I hope that did something to move toward an answer to your question. But essentially, yeah, it's an unanswerable question that is pretty nearly meaningless. And the more we focus on that, the less we focus on actually fixing the gigantic problem, which is SARS-CoV-2, which is everywhere. Yeah, and doing terrible things to everybody. And so I really, really like to keep the eyes on the prize and get rid of this virus. And then whatever, you can write, who shot JFK? COVID shot JFK. I don't know. Yeah, that's free. You can put that in your novel. Yeah, or somebody else can later on. I think that's all I got on this. Thanks very much. This has been Ask Dr. Ben.